welcome you all at Geolife Research Farm. In today's virtual meet, we will see in details about the different research activities performed by Geolife Research Farm in the different sections. The basic object of Geolife Research Farm is to conduct all kind of research activities in a scientific and precise manner. Secondly, to check the performance of each and every product before introduced into the farmer's field. So, for the perfect execution of product trials, we have a team of specialists from the different disciplines of agriculture, like major disciplines like entomology, pathology, agronomy, horticulture, soil science, even seed technology and agriculture biotechnology. So we do all the research activities at Geolife Research Farm in the different sections according to the product availability. At GRF, we have a 6 acre of land which has been divided into different sections, like here in-house lab trials, then plant protection trials like microfield trials, then pot culture and net house trials, in large scale area for the open field trials. In that we do foliar and fertigation trials in the nutrition segment. So every product undergoes for the 3 to 4 level of testing. Initially we do the in vitro lab trials for the plant protection products that may be insecticides, fungicides, then pot trials, then microfield and large scale field trials, as well as we also do multi-locational trials in the different locations to the different crops at different farmers' field. So how we do the research activities in particular areas, we will see in details in particular section. This is our agriculture entomology lab. All the in vitro activities that we perform here. So our entomologists work on this area. We'll see in details about how we do the research activities in an entomology section.
In the agriculture entomology lab, we are mainly working on 2 important aspects. First one is to maintain the pest population by artificial breeding, and second one is the in vitro efficacy study of different insecticides against different pests. In artificial maintain, we are mainly work chewing pests like fall armyworm of maize, diamond backmoth of cabbage or cauliflower, and Spodoptera lutura of cabbage and cauliflower on artificial diet as well as on natural diet. Spodoptera lutura mainly goes from different life stages. These are the eggs, these are the neonates, these are the first instar larvae, these are second and third instar larvae, these are fourth, fifth instar larvae, these are pupa, and finally the adult. And for sucking pests like aphids, acid, thrips, whitefly, mealybug, they require live host for their growth and development. That's why we are maintaining artificial chambers outside. In second objective of bioefficacy test, we can use 2 methods for bioassay test, like live method and direct exposure method. Here we can see the trials of aphids, Spodoptera lutura, mealybug, all the trials we can do in 3 replications, and after every 24 hours we record the observation, and based on mortality we prepare reports.
is our plant pathology lab. Here we perform all in vitro research activities. So let us know more details from our plant pathologist.
Jai Hind. This is a plant pathology section. Laboratory work is extremely important in plant pathology for initial screening of products irrespective to particular season. Research work conducted to be like isolation of plant pathogens, identification and its utilization for in vitro evaluation of product performance. Let's see first isolation and identification of different fungal pathogens. The fungal and bacterial pathogens can be isolated from different infected plant parts like leaf, stem, root, fruit. Here we have isolated 100 plus fungal and bacterial pathogens of different diseases like Mycospirilla, Fusarium, Rhizoptoria, like that. Thereafter use this pathogen for in vitro efficacy testing.
This is our pot culture trial section. So this is a very important section for primary evaluation of product performance in a short period of time. Here also we grow multiple crops to understand initial product performance with different dosage. Here we can see multiple crops that we have grown. One more benefit of this section is we can easily observe the root zone of the crop without damaging it, which is practically not possible in the field. Then next part, here we have the chambers for the sucking pest rearing like the dacids, aphids, mealybug that we maintain on live host in these chambers. So this is whole about our pot area.
This is our plant protection trial section. Here we conduct trials of various insecticides and fungicides based on standard protocols. Pest incidence and disease intensity varies from crop to crop and their growing seasons. Hence, to target different insect pests on the particular season, we have grown different crops like cabbage, chili, brinjal or also known as eggplant, pea, maize, cauliflower, okra, chickpea, so similarly we have one more plot of plant protection trial sections on the other side where we have sown the crops like cucumber, bottle gourd, bitter gourd, potato and tomato. The field evaluation using standard testing protocols is performed in comparison with standard check. Trials are performed in applications and the observations are recorded at specific intervals from randomly selected 5 to 10 plants from each replica.
This is the fertigation trial plot of cucumber. This plot is prepared for the comparative study of water-soluble fertilizers with 100% doses and nanofert series at a 20% doses. All the required nutrients and fertilizers provided to this crop is applied through fertigation system only. All the fertilizers calculated on the base of RDF. This fertigation schedule we follow for this cucumber trial. Periodically, we observe the morphological characters like vine's length, then number of leaves, number of flowers, then etc. The yield attributing characters we recorded, which is number of fruits per plant, weight of fruits per plant, yield per treatment, and yield per plant. At the end of the trial, the data recorded from this trial we use for the further statistical analysis as well as preparation of the final reports. We can see the comparative difference between water-soluble fertilizers at 100% doses and nanofert series at a 20% doses visually. Santulit Vedic farming is a holistic agricultural approach inspired by ancient Ayurvedic principles combined with the modern sustainable practices. It aims to balance crop nutrition, soil health, and pest management, ensuring higher yield and improved crop quality while minimizing the environmental impact. We have developed an Ayurveda-based comprehensive crop nutrition and protection packages, which is currently being tested against conventional crop management practices. Here we are conducting trials of a Santulit Vedic farming versus regular farming practices on a brinjal.
So this is all about the research activities that we perform with the help of our technical team at Geolife Research Farm. Thank you so much for your kind attention.